sort of direction, and he became one of the earliest adopters of high throughput sequencing for DNA metabarcoding in a sort of biodiversity application. And in 2012, he published this key paper that coined the term biodiversity soup. And I'm sure we'll hear about that a lot today. And the idea is you can sample a bulk environmental sample, sequence a bulk environmental sample to detect what's in it. And so since that time, he's been pushing methods forward and really linking DNA metabarcoding to environmental problems and policy. And so with that, I will hand it over to Doug.
Thank you very much.
And then just imagine the applause in the background.
Okay, all right. So you can all hear me, I take it? I am indeed, I'm going to talk about leeches, ponds, insects today. And I'm also going to talk about, weave it all together into something I'm going to call institutional innovation. And based on this tweet here by Ben Evans, who is a technology industry observer, I'm just going to show it to you here, and then I'll come back to it in the last part of the talk. So I work in China at Kunming Institute of Zoology. I also work at UEA, and UEA is a British university, which I'm going to say that I think probably all of you know, because it's not only a university, it's also the headquarters of the Avengers, which we really like. It's nice to have colleagues who appreciate field work. We can borrow their Mind Stone from time to time. And speaking of colleagues who invite like field work, here's a nice picture of Tal from when he was 12 or something like that. And he says I've mentored him, but I've just mentored him right here into getting his nose pierced with a needle and thread. So you're very welcome. Right, okay. Tal told me that the series is has a theme about beating the barriers in ecology and evolution, and about how I overcame barriers and so on. And I got a little introspective and thought about it, and I realized that I haven't beaten any barriers. I come from a very privileged background. It goes back quite a ways. My grandfather's uncle, my great granduncle, was Jing Si in the Qing dynasty. So it was some kind of high official. I don't really know. His nephew was got something like a university degree, Shoucai, and then my father got a PhD at Stanford. I got a PhD at Harvard. Now it seems like an unbroken chain. It has to be said though that this all survived the fall of the Qing dynasty, the war with Japan, World War II, Civil War in China, my father's statelessness until he made it to the United States. And I saw none of that, right? I just grew up in the suburbs, and I've been healthy my whole life. The most difficult thing I have to do these days is remote teaching. So basically, it's a good lesson here that privilege does persist across generations, even if money doesn't. And I've been a beneficiary of this. So let's make a big switch over to a little bit of molecular ecology as background. It's one of the things you can do these days is you can sequence a very, very tiny fraction of the DNA of an animal and bind that or link that to a Latin binomial, a taxonomy. And the most common version of that is this barcode, DNA barcode, which is in the CO1 section. And if you take a mosquito in my lab, here's a sequence that we got out of one mosquito. And you take that sequence, 650 letters, and you paste it into this web page, which is a public web page, NCBI, you get back this result, which is that the sequence matches at 100 or 99% to Anopheles quadrimaculatus. And it doesn't match, or it matches poorly, 92% and lower, to other Anopheles, and of course, much lower to other animals. So you can then say, this mosquito is a good match to this and a bad match to others, therefore it is in fact Anopheles quadrimaculatus. Doing this with one species is great, but doing this with lots of individuals at once is even better. And so the general pipeline, of which there are many, many variants these days, is to get some kind of bulk sample, which could be a bunch of insects, but also soil, water, feces, and so on. Lots of different code names. You extract the DNA. We don't do it with a mortar and pestle, but you extract the DNA. And then the main variant of this pipeline these days is to use a PCR to pull out one particular gene, to amplify it, make many, many copies of it, and then you send that to a sequencer. And then what you get back is just many, many, many copies of that one fragment of one gene, tens of millions, hundreds of millions. And so many of them are copies from the same DNA sequence, or from the same mitochondrion, or from the same cell, from the same organism. and from the same species. So you have to reduce this to clusters of reeds. So each one of these reeds now represents a number of reeds that are very, very similar. And the idea is that each cluster should represent a species. It's like a species hypothesis. So we call these operational taxonomic units, or OTUs. And then what you do is you try to assign a taxonomy to each one of them, like we've done here. Now, the thing is, it's not just one sample with many species, but in fact, you want to do those many samples. And so in the end, what you end up with is a table of samples in the rows, species in the columns with associated taxonomies. And as you can see, using this method, it's easy to add columns. It's also maybe a little less appreciated. That's also really easy to add samples, easy to add rows, because you're automating the taxonomy. So you don't have to worry about the fact that you have to deal with lots and lots of identification, but also because your samples these days, because you're doing DNA, can be easy to gather samples like water. So you can create these really high dimensional sample by species data sets. And the question is, what can you do with this? And very early on, when we were just getting started, we wanted to know whether this was going to give us, useful information. We're going to get signal from this. So we worked with a group of ecologists in Ailaoshan in China, and they were sampling moths along this altitudinal transect. So what they did was, the standard way of doing this, they pinned a bunch of macro moths, and then they took all the moths and sort of went around. I watched them go around and match them. I don't know how they do this, and they ended up matching to morpho species 8,000 specimens. And we said, could we have some of your samples? I said, oh yeah, we got lots. So we just gave a bottle from every sample, and then it took us half the time in person hours. This is when we're just getting started too. And we could do all the arthropods in the samples, not just the macromoths. And then we compared results. And so you can see in their study, they were able to differentiate altitude. So every point here is a sample, and if the points are close together, that means that the samples share a lot of species in common. And so you can see that the samples cluster by altitude, and also a little bit by ground and canopy, whether the trap was in the canopy or in the ground. And then these are our results from the time. And we also can differentiate altitude, and we can also separate ground and canopy. So it looked like it was really working, and we did this many times, and many other people have done this many times. So it really seems to work very, very well. So the question is then, now that you can do this, and you can automate a large chunk of ecology, what can you do? So one of the first questions that we came up with was, we wanted to measure the effectiveness of a nature reserve, right? And just in this last year, of course, we have a secondary question on top of this, because China and Vietnam have banned the wildlife trade in their respective countries. So how good is this ban? And will it have an upstream effect to finally reduce poaching? Is that actually going to happen all the way up? And so the thing is, it's really, really hard to know whether a nature reserve is working. You can measure forest cover, you can measure management budgets, but does it actually protect the biodiversity inside the park? And Maxwell et al. Last year conveniently published this review, and their answer to this question is, we don't know. All right, the world does not yet know whether or not nature reserves actually have positive biodiversity outcomes, which is a terrible thing to have to conclude, but it's true. Okay, so why? Because it's really difficult to see animals, see wildlife. I mean, this is a picture taken by Andrew Tilker in Vietnam, and there are two large mammals in this photo. You may or may not be able to see, there's one right in the center, light colored. It's a human being. So it's a trick question. There's another human being to the left. Those are his team members. You can barely see your own team members. How can you see wildlife that are trying to hide from you, or nocturnal, arboreal, and so on? So we went to a nature reserve the size of Singapore in southern China. And you can see it runs along a mountain ridge. And we wanted to ask, where are the wildlife? Where are the vertebrate wildlife along this park, right? And which species are they? And so we wanted to capture the DNA of these animals. And so this is what I mean by you can get lots of samples. Instead of collecting the DNA directly, we use a professional collector, leeches. And what they do is they feed, they're full of blood, they go off and digest for a while, they shrink down again, and they come out for their next feed, but they still retain a lot of RNA and DNA from their animals. And then you can collect them and then do PCR on them and get something out. So what we did was we contracted 163 rangers to go to their patrol areas they're mostly checking for forest fires and so on. And they collected for us 30,000 leeches in 3 months, spread over lots of little tubes of RNA later. And then just to jump ahead for a second, we then took those and we found everything from frogs to snakes to reptiles to birds and lots of mammals. Okay, now when we're doing this, one of the things we realize is that a ranger going out is patrolling. We can force them to take replicate samples by giving them these little baggies, little Ziplocs full of tubes. Each bag is like a little replicate. So that's exactly what you need for occupancy modeling. And you don't have to train anybody to do it. You just give them bags like this. We designed this long molecular and bioinformatic pipeline biased towards getting rid of false positives. I'm not going to go into the details of this other than to say that if you do something similar to this, I would suggest that you try using this software called ProTax to help you assign taxonomies. Okay, and then apply the multi-species occupancy model, because even if a species is present in a site, you might not pick it up, right? The typical false negative problem. And so in our particular case, our multi-species occupancy model, our ecological process, it modeled the presence or absence of in each species by the elevation of the site and by the distance to the reserve. So those were our environmental covariates. And then of course, you might get, the species might be present, but you might not detect it. And our covariate for that is the number of leeches in that sample. So kind of our sampling effort, right? And so this is really important because we don't need to rely on rangers to exert effort and attention while they're going out and sampling, or we can just count the number of leeches that they actually collect. And so one of the places we fell down was in getting metadata back. We got a lot more pouches back, but we weren't great at getting the metadata back. So we ended up getting about half of the samples actually came back with metadata on site. So we've now corrected this and figured out how to do this. But luckily, when you do modeling, you can interpolate across sites because of the environmental covariates. So this is the interpolation of the model, and you can see that one of the first results is that there's higher species richness towards the center of the park and towards the south, which is the more tropical part of the park. Now I'll show you two maps contrasting. This is the muntjac, and it's found more at lower elevations than towards the north and towards the edge of the park. And then there's a leopard cat, and you find it more towards the center of the park at higher elevations. If you look at this overall, the species in the data set, and we can divide by warm-blooded versus non-warm-blooded, you can see that there's an effect of elevation. So occupancy increases in general with elevation, which is not too surprising. But when you do this by individual species, all those little lines, right, you can see that most of the species show this positive. Some species show nothing. That's mostly frogs. And then a few species show a negative relationship with elevation. They found at low elevations like that muntjac. Distance to reserve edge, it's a much weaker effect. And it's partly because elevation and distance are correlated with each other. And we can ask, what are the species that show this negative relationship with elevation, that are ones that are found near the edge of the park and at low elevation? Well, 3 of them out of these 5 are domesticated animals. And we know from camera traps and just know that there's a lot of grazing of domesticated animals around the edges of the park and in the park. The other one's the muntjac I just showed you, and then the last one is the Rattus nitidus, Himalayan field rat. And muntjacs, and obviously the rat, are resistant to hunting, and we know hunting also occurs in this park. So it seems reasonable to conclude, or to at least strongly hypothesize, right, that human disturbance is driving wildlife up into the higher elevations. Okay, so we can't answer Maxwell et al.'s question, for all the world, but we can answer the question, does this nature reserve have a positive biodiversity outcome? And the answer is mixed, right? So there's literal room for improvement. There, we can see where the wildlife are. We could go back and check with more sampling and with other methods if we want to, and we are doing that. I think on top of this, and this is really important, is that the data here and the method here have some very important and useful properties, which we don't usually think about when we're trying to do work. So first of all, the data are efficient. We can get samples in 3 months. We've done the other thing. It takes about $10,000 to get 30,000 leeches. It's nothing. It's repeatable because we've automated the process. It's direct. We're directly measuring wildlife, right? We're not measuring management budgets, right? This is a point that Maxwell all made. It's granular. We're measuring individual species. We're measuring individual sites within the park. And it's understandable to public. You can just show maps, and it's auditable. It's possible to go back and check the results that are here using camera traps or direct visual censuses once you have a general idea of what is apparently where. So I'm just going to stop right there, but I want to thank Yin-Cho Ji for the devising the molecular and the bioinformatic protocols, Chris Baker for doing the multi-species occupancy modeling, and Wenqing Zhong for he works at the Yunnan Institute of Forestry Planning. And I gave a talk in China, and he says, you really should do this leech thing in a real park. Why don't you come do it in Ilao-Sen? And it's great to work with someone in an actual reserve. Okay, so here's some photos of Chris and Yin-Cho. Right. Okay, so that's question number one. So let's go to question number two, right? Where is the vertebrate biodiversity in a mixed-use landscape that's not tropical? And so this is Middle England. This is known as the Southern Midlands. And we want to measure the vertebrate biodiversity around here. There's a lot of semi-natural habitat in England. It's very agriculture dominated, but there's still a lot of semi-natural habitat. And we don't have blood-sucking leeches to help us do collections. They're all in the House of Lords. So we have a big sampling problem, right? It's mostly private property. We can't get onto the land, and it's just a huge amount of effort to be able to cover something like this size of area, never mind all of England. And of course, there are lots of domesticated animals as well. But what we can do is we can take advantage of a DNA sampling program that already exists. And that is that in the UK, the great crested newt salamander lives in ponds, breeds in ponds. And if you build next to a pond within 500 meters, you have to do a survey of that pond. So it used to be these visual surveys, send colleges 4 times every pond, and then they would count. Nowadays, you can do a water sample, send that to a specialist laboratory. They do qPCR, and they tell you whether the newt is there or not. And if it is not there, you just go ahead and build. If it is there, you have to do translocation. Builders hate the great crested newt, not surprisingly. So on top of that, another company showed up called Nature Space Partnership. I'm not involved with them. And what they did was they said, instead of ad hoc doing a sample every pond every time you want to build, let's do 600 ponds across this area that I'm showing you here. And then we can take those 600 ponds and turn them into a species distribution map. And then after negotiation with local government, they turned it into 4 categories: white, green, amber, and red. And if you build in one of the green, amber, and red zones, you pay some money. And then that money does not go to surveys, it doesn't go to translocation programs, it goes to building new ponds. So they create new habitat. So this is, I'll come back to this company, but for the moment, what we got was basically free samples. A lot of the DNA had already been used up, but we got about 300 of these ponds. And I very roughly estimate it was like $200,000 if we had wanted to do it. And it's really impossible to do. They had like a full-time person calling landowners, asking permission to go onto their ponds. So these samples went to Taiwan, my PhD student, and she metabarcoded it, and so got 47 vertebrate species, and the more common ones, everything from birds to mammals and so on. So the way you think about ponds, right, is that there are toilets for birds and mammals, and they're capturing their DNA, and then of course fish, amphibians live in them. And then what we wanted to do is we wanted to make some sense of this, to model it, and we are using a statistical technique called joint species distribution modeling, and we're using a package called SJSDM, which I don't think is published yet, but it's available in preprint. It's certainly available on CRAN. And luckily, every time you do a survey of a pond, a professional ecologist does a bunch of measurements that are very standardized in the UK, like pond area, drying, like guest drying frequency, and so on. And then we can download the land cover map for the UK and get some land cover variables. And if you do that, you can start getting a view of the niches of the different species. So I'm just going to look at the 5 amphibian species here. It turns out the smooth and great crested newt like smaller ponds. They have a negative coefficient here. They like ponds with heavier macrophyte cover, which we know has to be true because, among other things, they lay their eggs in macrophytes, and fish that eat them also eat the macrophytes. So that's kind of a proxy for fish. And then the other 3 species, which don't really show any preferences for ponds, per se, they show preferences for the land cover around it, woodland. You can do this for all the species times all the variables, right? But we won't. But the point is that it's possible to now map the vertebrate biodiversity of this area using an already paid for sampling method. Okay, so question number 3. How will biodiversity respond to climate change, right? So I'm not a climate change biologist in any means, but let me just very quickly describe what other people do, which is that, you have the most frequently used method for trying to answer this question is that you have a climate map now, you have some projection of the climate in the future, you have a species distribution map that has climate variables in it, and as the climate changes, you infer that the species will, if it's possible, change its distribution along with the climate, maybe moving north, maybe moving upwards, and so on. So this climate correlative approach is the most frequently used approach, but everybody knows, right, that it ignores a lot of biology, and in particular, it ignores species interactions. And that's for a good reason, right? We don't have a lot of information on species interactions. And tied up with this is the observations that are being replicated now and, under debate, but that insects seem to be in heavy decline, right? So something with global change of different sorts, insects seem to be in decline. Okay, and there are multiple papers, and I just want you to look at the lower one here, ecography paper. This is taken in Greenland. There are no human populations at this site, and over time they've seen an 80% abundance decline of muskid flies. So this doesn't seem to be like a direct human thing. Okay, so I was very fortunate to be invited by Thomas Roslin to join his project Arctin, which has used samples of arthropods that have been collected at Zuckerberg Research Station by Niels Martin Schmidt, who runs the station and runs all the biology that comes out of there. And it's a great place to work, right? Because it's got a very simplified ecosystem compared to the tropics. It's mostly flies, wasps, spiders, right? And a few vertebrates for food for the insects. And they've been captured. Niels has been organizing, running the capture of these using pan pitfall traps since 1996. And they're brought back to Denmark. They're sorted to family level and counted and so on that way, but that's it. And then put into jars of alcohol, and they've been sitting in a warehouse in Denmark. And so here they are. And so we're talking about 375 above ground arthropod species, and they had like at the time we were doing this about 750,000 individual bugs, right? And so we were able to get of this 375 about 300 mitochondrial genomes assembled that we did, and then we took those, we reconstituted those trap samples, and then just shotgun sequenced, which means we just sequenced all the DNA, of which 0.1% or less is mitochondrial DNA, and of which tiny bit is the CO1. So we made a lot of libraries, spent a lot of money doing this. But then what you can do is you can take those reads and you can map them to the different mitochondrial genomes. So here's a mitochondrial genome on the top. And there's all those little green things are reads that match them at high quality and don't match other genomes. And so we can say that species is present at high biomass in this sample. And then you might get something like this. This is a lot of reads are attracted to this genome, but we can very confidently say that that species is not present because we should expect reads across the entire genome. We're only getting reads at a section, so that's a conserved site to false positive. And we've done tests, and it really does a great job of taking false positives out. And so when you do this for many samples, and then of course you get many species, you get what I call a community time series, right? From 1990, we did it from 1997 to 2013. So I'm showing you just a small portion of the data set, so you can just have a look at it, right? Each row is a species, each column is a week. And red means that you get a lot of DNA, so a lot of biomass. So it's like an activity or phenology peak there. And it's important to notice here that summer has doubled in length during just this small period of time. So climate change is going faster there than it is where we live. And if you sum up those data to get some summary information, you see insect decline. The observed species diversity richness has declined from by half during that time, and temperatures increased by 2 degrees. So it really is like a laboratory of the future. Okay, so what's going on, right? Can we figure out what's causing this? what's implicated in this. So in a climate correlative approach, you would expect to see shifts towards earlier dates in activity peaks, right? So here's the species in blue that shows an activity peak in August, and it doesn't show an activity peak in August in 2013. It doesn't show one, sort of smeared around July. Okay, so that kind of makes sense. That's a climate, it's a climate envelope following the climate envelope. But here are 3 species that show the same kind of peak in August, and they don't show any kind of peak in August, and don't show any peak at all. It's kind of is petered out, disappeared. In fact, there are lots of species in the data set that are like this. They show a peak sometime in 1997, and they don't show something in 2013. And a lot of species that show the opposite. So species that were like constitutively rare, we hardly picked them up in 1997, and then they show these big peaks sometime in 2013, right? None of this stuff seems to be easily explainable by response to just increasing temperature. So this is just a small portion of the data. So I'm just getting used to what it looks like. But if you want to actually model it properly, the first thing to do is to work with some very smart people. So we were really fortunate to work with Nerea Abrego and Otto Vizcainen, who literally wrote the book on joint species distribution modeling. And what they did was they modeled the presence or absence of a species in a week by that species, the previous month's temperature, the week in which that sample is from, the week squared, so you model up and down for species phenology, and then species interactions. So species interactions being intraspecific density dependence, all of last year's, for this species, all of last year's incidence is summed up, right? So that's like a measure of last year's abundance. You take all the herbivore species in the data set, you take all their incidences, sum them up, that's all of last year's sum, that's the herbivore abundance last year, parasitoid, predator, detritivore. So a really simple way of summing up like the last year's other species and how abundant they were, and divided by trophic group. Now we've done this lots of different ways, but this is just one way, just showing you this. And then of course you do this for each of the species that has a lot of incidences. So we're talking 28,000 data points in this data set. And then you compare the performance of two versions of this model, one with just climate and week, right? And then one with climate, week, and last year's abundances. So that's the biotic model, the more complicated model. And you ask, how well do they fit to the data? And how well do they predict the data? So we can, you can measure the explanatory power, how well they fit the data. And what you find is you can compare the two. And what the 1, 0, and -1 here means that 1 is like a perfect prediction, 0 means random prediction is just doing no better than random, and -1 means it's worse than random. When the species is there, it's predicting it's not there. When the species is not there, it predicts it's there, right? So that would be worse than random. And if you look at this, the abiotic model, that's the simple model, they all do, actually both models do pretty well. The abiotic model does better than random for all but 4 species. The interactions model, the more complicated model, does better than random for all the species. And in fact, just generally does better, right? It's got a better predictive explanatory power. But okay, it also has 5 extra terms, right? So it could be fitting noise. So the real test, right, is to fit the model to 14 of the years in the data set and see if it can predict the 15th year holdout when you do this for all the years. And what you find, of course, is predictive power is lower, but the abiotic model is terrible, right? So it's random or worse than random for 80% of the species. It only does a reasonable job for 20% of the species. The interactions model, more terms, so therefore more prone or more risking fitting to noise, it does better than random for 50% of the species. So it does better. And there are different versions of this model and so on. But the same result comes out through, right? It says that the interactions model does a better job of predicting. So it's this is primarily a statistical exercise using, and we going all the way from very far-sighted, like a really genius move, I mean, in my opinion, to collect systematically insects starting from 1996 all the way through, then finding a way to pull the DNA out and create these large data sets, and then to analyze it with joint species distribution model, which didn't even exist as a method when it got started, right? And what is the answer? And the answer is we don't really have a great answer other than biodiversity response does seem to be, in this data set, in this place, strongly affected by species interactions. And we were able to infer this from a set of data poor species. And so I think this is kind of one of the opportunities that we have with these new methods, that we can create these community time series. Okay, I've been told that there are a lot of students, grad students and postdocs watching this, so I thought I'd talk a little bit about institutional innovation, where I think I see ecology and environmental management going in the future as a result of these technological changes. And it's also a shout out to Agents of S.H.I.E.L.D., which I think is just a fantastic television program. Okay, so if you're managing your education and you're interested in trying to figure out where you're going to put your effort, and you can think of your skill sets as being in 5 different boxes. And I think it's it's unrealistic to learn all these boxes well, right? But it's realistic to learn 2 of them, and then to also learn how to work with people in the other boxes, right? So the blue here represents what I kind of think of myself. I'm a field biologist by training, and then I just added like lab and analysis, some lab and some analysis to be able to work. And then I try to work with people in the other boxes as much as I can. And if you can do that, right, or groups of people can do that, you can create a flywheel that links all these things together and achieve something I'm going to call institutional design or institutional innovation. Now, what's an institution? Institution is just a set of rules that get a bunch of people with lots of different motives to work together towards some common purpose. And we're constantly trying to figure out how to design institutions. Professional sports is a real-time institutional design problem. And so is conservation. And the Nature-Space Partnership, which I mentioned earlier, is one of these institutional innovators. They thought, instead of doing these ad hoc surveys, we'll do 600 systematic surveys and then do some negotiation. And if you build in a green zone, you pay between 0 and 50,000 pounds. It's about $70,000. If you build an amber zone, 0 to 90,000 pounds. If you build in the red zone, you pay 0 to 100,000 pounds, right? And you can do this at any time of the year instead of being limited to the breeding time, because you just have to see where you are on the map. And of course, the size of the amount of money you pay depends on the size of your development, where you are located exactly. And then that fee then goes to building 4 ponds per effective pond. Actually, what they do is they build ponds and then they cross them off for each one, and then that fee goes to build more ponds for the future developer. And then they also endow a long-term management fund. So they've created a biodiversity offset business that changes the way that it works. So the whole story looks something like this. The UK government first passed a law to protect the great crested newt, and then they passed subsequent rule changes to allow eDNA sampling to take over visual, and they allow this district level licensing biodiversity offset going on. Then Nature-Space Partnership then contracted Nature-Metrics, another company, to do this one-time eDNA survey, which was not possible to do under visual surveys, but it became feasible with eDNA. Then they parameterized a single species distribution model. And then they did this, they built a business. They spent something like £1 million getting started. And this negotiating, marketing, websites, legal, and building ponds. And so that's funding and scaling. That's what I mean by funding and scaling. They built a whole new way of doing things. And in the last 2 years, they've raised about £1 million, £1.3 million in US dollars in these fees. They've created over 100 ponds already, and they've endowed a 25-year management fund to manage those ponds in perpetuity, well, 25 years. Okay, so I promised to talk about this tweet. So this Ben Evans is a technology observer. He has nothing to do with biology, but he has this really great summary of how technology affects us. So when technology is introduced into some industry, the first thing you do is you just use that technology in the old way of working, right? So eDNA became possible, and instead of changing anything, instead of doing visual surveys, you just did a DNA survey. All right, but you still had to do it in the 3 months of the year when there was breeding. And if you did detect GCN, you still had to go and translocate them. So nothing really heavily changed, but it was faster and cheaper. So people took it up. And then Nature-Space Partnership, that's the next stage. Once you understand the technology, you change the work to fit the technology. You really understand the technology and all it can do, you change the work. So once you do this, this map, right, you can you can give a license out at any time of the year, not just during breeding season, right? It funds habitat creation rather than translocation, which only at most benefits one species. And really importantly, it de-risks construction. It's a terrible word, but it means that if I'm a builder, I just know how much I have to pay, and that's it. I'm not gonna be delayed by a year while you're doing translocation. And that reduces the political pressure against protecting great crested new, which I think is very important. And so there's one takeaway message, right? The long-term benefit of technology is to help you and us facilitate innovation in the design of institutions for biodiversity conservation. And we have to think about it in those terms to really take full long-term advantage of technological innovations. And I mentioned Nature Metrics. It's a company that provides environmental DNA technologies to industry, to governments, to NGOs, to academics sometimes. And it's a company that I co-founded in 2015 with Kat Bruce, Alfred Vogler. We got money from the government, we got money from my university here, and Kat ran it, CEO for 5 years, finally stepped down after 5 really busy years. And one of her achievements was to get a Series A funding round in 2019, which started a growth phase all through last year and this year. And it currently employs, I think now over 40 employees. I mean, this was prepared a couple of days ago, and then I think they've just hired 2 more people. So, and just about half of them have PhDs, and they work around the world. They work on samples from everywhere with large companies, and they're proposing to get bigger and bigger. And the idea is that they are providing the technology for clients who can't do eDNA, and then they're building an analysis team, and they're also starting to work with clients to work on funding and scaling. So trying to get these flywheels going in as many places as possible. They don't do policy and law, but the idea is you work with policy and law that exists and try to make policy and law, adjustments if it's possible in various places. But the long-term benefit of technology is to try to facilitate innovation in the institutions of conservation. Okay, thank you very much.
All right, thanks, Douglas. All right, thanks, Douglas. Anybody who has questions can post them in the Q&A tab. I'm having some Zoom issues here myself, but feel free to ask anything that comes to mind.
Where do I find the questions? Do I stop screen sharing? Oh, there you go.
You can stop screen sharing. I'll also note any questions that come in.
Okay.
I'll ask one of you, Doug, to break the ice here while questions come in. What do you see as the future of using environmental DNA for environmental management beyond what you just laid out with transforming institutions? How are technological changes going to influence what we can do?
I think one of them is auditing, make it possible. I think in general, we have to assume that most people don't believe that most conservation actions do anything, right? And how do you show that? And so information evidence needs to be what was often called third-party verifiable, right? So it's not just that I know, and not just that you and I know necessarily, but anybody could check. And so camera traps are a really good example of that. Bird lists, where someone says, I saw this bird in this place, absolutely not that, right? And so they're good for science, but but trying to get third-party verifiable evidence is really tough. And eDNA has that possibility because you can allow different people to take the same samples in the same places, and they can be sent to different labs, competing labs. So I think that's really important. And also, I think the other thing is that it's going to allow us to take time series much more easily. So what we're finding is, for example, companies that mining companies and so on, they send in groups of biologists to do an environmental impact assessment, and then they leave. And then they can't see change because it's too difficult to have a team down there all the time. It's impossible, basically. But now they can leave sampling kits down there with someone semi-trained. They can take samples over time, right? And then they get change over time. So time series and effects of the environment is another game changer. I think those are 2 really important things.
All right, the Q&A tab is starting to fill up. I'm going to start with a question from Lou. It's a multi-part question. It says, super cool talk. You've worked with these organizations that have already collected samples, organizations that did not know what your research question would be at the time of sample collection. Do you wish they had done anything differently when deciding what, how, when to collect samples? What advice would you give organizations collecting data and samples for future use by the scientific community?
Oh, that's a very simple question to answer. So the way they collected it is water into tubes with ethanol in them. And this is because the very first time they tested this, they used that. And then the regulator said, you have to follow that method. That's a terrible method. It's like the worst method for collecting water samples. So the answer is collect water samples into water filters with some preservative fluid and take multiple samples per pond. Right? And that would allow you to do occupancy correction and also get you much more DNA per pond. So in general, for water samples, filters are the way to go.
All right. Brooke is asking another two-part question. First part is, what do you think is the best pipeline for species occupancy using metabarcoding data? And the second part is, when do you sleep?
Oh, I sleep a lot. I sleep tons. So the best piece of career advice I ever got from John Terborg was change the thing you study every 5 years. And I took that to heart, right? So what I do is I work on something for 5 years, and then you're not really writing papers on it, but eventually you start writing papers on it. And then you kind of write papers on that subject on and off for a long time afterwards. So it seems like you're working on lots of different things, but actually you worked on it for a while, right? Metabarcoding, all this molecular stuff, is the first time I've really worked on anything for like 10 years. It seems like a really long time now. But that's the way to do lots of different things. And then you're fresh, right? Pipeline for species occupancy using metabarcoding data. So metabarcoding data has the cost that it also has a lot of false positives and positive detections. And so the most of the answer is that you have to bias the metabarcoding pipeline so that it reduces false positives, even if it causes you to have more false negatives. I think that's the way to go now. One of the reasons we use that whole metagenome thing was that we were able to just cut out false positives using that percentage cover filter. For PCR, it's much more difficult, but there are methods. But that's where I would put my attention, on the metabarcoding pipeline. Occupancy modeling is still open. It's an open area of open research, I think.
Okay. Cheyenne asks, what environments do you think it may be difficult to use eDNA in, or where eDNA may need an additional surveying method accompanying it?
I think they all do. They all need additional survey methods, partly because when you're first doing it, obviously, at any given location, you need some kind of validation. And then also, I think our eyes are really important, right, to be able to see things. So vertebrates are particularly difficult. I will say that there is a preprint out for those same leeches can also be used to sample the viral diversity inside those same animals. So it's possible to get the viral diversity from wild animals in the wild without actually capturing them. They're mostly ground-dwelling animals. I don't know what would be complementary to that, because the only other possibility of doing that, the only complement to that would be like capturing the animals themselves, which I think is just crazy. But camera traps, I think, is a really great complement for a lot of vertebrate eDNA work.
Okay. Matt is asking, are some of these models capable of handling microbial assemblages? Do the models take the OTUs and their abundance directly, or do you form the models after you understand the community?
So it's almost like the eukaryotic and microbial metabarcoding and other methods, metagenomics, have evolved really separately. So for ordination, for example, my understanding of single cell and metagenomics is these methods like TSNE and so on that I'm not aware of. I don't know how to use. JSDMs, joint species distribution models, I think right now are purely used by the eukaryotic people. I think I think in general, they're scalable. The JSDM, the reason we're using JSDM is because it works on lots of rows and lots of columns. It's a GPU-based JSDM method. It's hard to use something like HNSC on that. And so I think it's very, very transferable. One of the main issues with all these data, right, is that you have row noise and species biases. And so you hear people talking about you can't really count abundances. It's not true. You can do abundance. You can get abundance information out of this. We've shown it experimentally, but you need to do things to your pipeline. You need DNA standard spike-ins. You need to do maybe whole genome long read sequencing to do relative species frequencies. I mean, it's very doable to get abundances out. And with microbial stuff, that might be less of an issue. But I'm not much of a microbial person, so I can't tell you fully.
All right. Ivan says, great talk. I'm wondering if by comparing 2 years, you'll see a shift in phenology given the interannual variability in climate. If you don't have a long enough time series, maybe you can try to use extreme years, e.g. drought versus wet year.
More data are better. So they only started in 1996, and really the samples were partial. So we started 1997 for a full sample. As it happens, the 2010 samples never made it back from Greenland to Denmark, and so they got lost. And then they were found again last year in Cambridge, UK, and returned. So we're hoping to sequence them, and then at some point in the future, we could sequence the rest of them, get the 1997 all the way to 2020 samples. I don't know if they did 2020 samples for obvious reasons, but you can only use what happens in that time series. Right? We're not making it anything happen. There are other habitats, other trap lines. So maybe that will. Some of them are wetter and some of them are drier. This is like population ecology where you're looking at time series and trying to see what happens, but you can't. They're natural experiments, right? So we can't really. We can only hope for extreme events, basically.
Yeah. Somebody who I think has named themselves after their camera is asking, how affordable is eDNA for the average private consultant? In other words, is eDNA affordable for all sectors or just large NGOs at this time?
So the United States, I think you're going to pay somewhere around $100 to $150 per metabarcoding type sample. For qPCR, it's less than that. It's somewhere in that range. And so when a business is doing this, right, they are investing in sample tracking software, billing systems, constant, having lots of different samples so they can constantly be running things through sequencers, and where there's ability to if you're interested in that kind of thing, where there's ability to negotiate lower prices, one of the things you can do is say longer turnaround time, and then if someone has extra space on a sequencer, they can fit that stuff in. So that's one way to really reduce costs. Another place to really reduce costs is in doing the DNA extraction yourself, if you're able to do that, because that's sort of the most time-consuming part. And I'm just giving you like specific information on how to get prices down, but those are the two main ones.
All right. Daniel says, amazing collection of projects. Did you find it challenging to juggle co-founding Nature Metrics with all of your academic and research responsibilities? What advice would you have for people trying to bridge the private and academic sectors?
Do one at a time. So my role, okay, so when we did this, we had the academic consultancy model in mind, and we had the startup business in mind, right? And to Kat, who did a PhD with me, we discussed this a lot. And then when she graduated, what she first did was go work for an incubator, a small business incubator, right? And she just kind of left science for a year, and then we got back in touch at the end of the year, and I said, "Do you still want to start a business?" And she was being headhunted by different startups and so on. I said, "Okay, no, let's just start a business." And at that point, I focused on helping to write the grant proposals for government money. It was a small amount of money. And then she focused on getting money from angel investors. which are people who put money in a very, very early age, very early stage. And then once it started, hired people, and I did not do anything after that. Like in the sense of like, I'm there all the time, and I'm on their teams and so on, Slack and so on, but I'm not involved in the day-to-day management because you can't do a good job unless it's a full-time job. So we made the decision that it would be a full-time thing with actual staff. We would run it at that level, fund it at that level, try to grow it to that level. There's no way to otherwise separate your minds. You can do contract work as an academic, but this is a different scale, right? So so that's how I do it. And then sometimes I mean, often I see a paper, I see this and stuff like that, and I'm constantly talking with them about statistics and new things, new ideas, but not management.
Okay. Marie has a question.
Hi, Marie.
Yep. How big is a leech home range? Is there any way to correct for larger home ranged inverts or insects that sample the vertebrate population?
So we don't know about other insects, but the relevant home range is the distance from the time it drops off after having fed to where it's captured next. And so you can imagine like a blood-fed mosquito, a blood-fed fly, a blood-fed, well, let's just stick with blood-fed mosquito, a blood-fed leech. They're full of blood. They probably don't go very far, dig in. And so the next time you see them, probably pretty close to where they were dropped off, we think. The only possibility is that they might have ridden on an animal and then been taken off before they could feed. So they still have like 2 animals to go. That would be a way you get a really long dispersal event. But with things like blood-fed mosquitoes and blood-fed leeches, we think very close to where they were dropped off. The main issue is how long ago, right? Because you might pick up a leech here, it was dropped off here, but the animal's long gone, right? And so we can only estimate what that is, like because the blood lasts there for in a leech for months in laboratory tests. Now, when it comes to like flies, we know that they follow along, right? So in that case, the home ranges are probably quite large. Probably like they follow the home range of a whole monkey troop, for example, and just feed on feces as they're going along. That's probably the conservative answer for them.
Okay, and a follow-up question. How do you account for that lack of a specific trap location when you start plugging this data into occupancy models?
It's the patrol area. So that patrol area is like a whole day's, walking. So it's 173 patrol areas in an area that's 677 square kilometers. So our assumption is that that the animal is in or near each of the patrol areas where that leech was picked up. And we can't get any better than that. Luckily, the answer we get that elevation is the primary, environmental covariate is reasonable, something that we can check and something that we intend to check.
All right, PixelXL has identified themselves as Morgan and is asking, would eDNA be applicable in studying soil microbial biomass in the rhizosphere, or is this tool specific for aquatic systems only?
eDNA in the broadest sense of DNA from the environment, not DNA that's trace DNA, that's released from the environment. Yeah, sure. In fact, one of my colleagues here studies microbes in rhizospheres in the root and next to the root, and they use amplicon sequencing, but they call it amplicon sequencing. They can call it metabarcoding, and they don't call it eDNA because that's not part of the microbiology world. But yeah, absolutely. In fact, most of our methods come from microbiology.
Awesome. The Q&A window is currently empty.
Thanks, Doug.
Thanks
for your time.
Amplicon sequencing.
I don't want to keep you too long, but I do have one more question for you.
Sure.
A paper came out suggesting that soil was a good source of eDNA to detect vertebrates, but you had suggested before that it probably wasn't, and we've been doing soil cores and haven't been detecting lots of vertebrates. Do you know, is there consensus on detecting, like, say, forest vertebrates by detecting soil?
I think it's really tough. One of the reasons it's really tough is that you get so much high quality DNA from fungi and particularly microbes and so on that even if you're trying to use specific primers, it's going to be really tough to do. And also there's a lot of degradation. There's a lot of things trying to eat that DNA. But my recollection is this was in Jasper Ridge. Is this the paper in Jasper Ridge?
Yep.
So these were camera traps. These are soils from sites where they had camera traps next to them, and so along tracks, right? So maybe those sites have a lot of urine and so on. I do remember one of the very first papers I tried to do it in the zoo in Copenhagen, and they were able to get the DNA from the animal in the pen, in the midden, but not on the other side of the cage or the other side of the enclosure. So I suspect this doesn't work terribly well. What works okay actually is pond, like water from water holes, and my guess is clay licks would probably do the same thing.
I have one question for you. Obviously, all of these eDNA techniques are founded on the shoulders of classical taxonomy. That had to be done at some point. Where do you see the future of taxonomy in the sort of wheel of the eDNA pipeline, and where does it fit in, or does it fit in?
Oh, I think it's really important. I mean, every OTU that we are unable to identify the species because the reference isn't there is lost information. So, we want to know why species are there, and the way you do that is by correlating traits with their distributions. And if you don't have a reasonable taxonomy, it's very hard to get the traits out. And okay, so I work with a lot of arthropods and so on, for which even if you knew their name, it's very hard to know what their traits are. But I think it's deeply important. So luckily, there are lots of efforts to sequence everything on Earth. Because if you're a genomics person, you don't really get involved in conservation the way you get involved in conservation is to sequence everything on Earth. But it happens to throw off a lot of useful information. So I'm just hoping it just continues and builds. And so, a lot of conversations in the United Kingdom are priority setting, like which group should we sequence, before other ones. One place I see where we will kind of never really get Latin binomials is soil fauna diversity. I think we're always going to be stuck with sort of higher level taxons and just huge numbers of OTU numbers or bin numbers or something like that. Nematodes, for example.
Thanks, Doug. We should let you go to sleep.
We're going to go to McDonald's or something. I don't know. It's just like anything that's open, just to kind of live a life, right?
Well, thanks for coming, everybody.
Thank you very much.
Thank you.
I'll make sure to have an embarrassing photo of you when you invite me to give.
Excellent. Okay. All right. Okay.